Incredible breakthrough: tardigrade protein protects human cells from radiation. Date: March 5, 2025. Summary: recent research has uncovered a groundbreaking application for a protein derived from tardigrades or water bears which can protect human cells from radiation damage, particularly in the context of cancer therapy. This study, conducted by researchers from the Massachusetts Institute of Technology, MIT Brigham and Women's Hospital and the University of Iowa, was published on February 26, 2025 in Nature Biomedical Engineering. The findings herald advancements in cancer treatment, potentially extending to astronaut safety during space missions and longevity therapies. Background on tardigrades: nature's extremophiles. Tardigrades are microscopic creatures renowned for their extraordinary resilience to extreme conditions. They can survive environments with radiation levels that are 2,000 to 3,000 times greater than what humans can endure. This remarkable ability is largely attributed to a unique protein known as Dsup damage suppressor, which binds to DNA and mitigates radiation-induced breaks. The challenge of radiation therapy in cancer treatment. Approximately 60% of cancer patients in the United States undergo radiation therapy, which, while effective at targeting tumors, often leads to debilitating side effects. These can range from painful mouth sores to gastrointestinal damage, significantly impacting patients' quality of life and sometimes forcing them to halt treatment. Given the urgency to address these side effects, researchers sought innovative solutions inspired by the protective mechanisms found in tardigrades. Harnessing Dsup for human applications. The research team aimed to leverage the protective properties of the Dsup protein by utilizing messenger RNA mRNA to induce temporary expression of this protein in human cells. Unlike direct administration of the Dsup protein, which could elicit immune responses, the researchers created polymer-lipid nanoparticles designed to deliver mRNA encoding the Dsup protein to specific tissues, including those commonly affected by radiation therapy, such as the oral and rectal linings. This method allows for localized production of the Dsup protein, providing a protective shield for healthy tissues while ensuring that tumor cells remain susceptible to radiation treatment. Key findings from mouse trials. In preclinical trials conducted on mice, the researchers observed a significant reduction in DNA strand breaks, up to 50%, in tissues treated with the Dsup mRNA before exposure to radiation. This promising result indicates the potential for minimizing collateral damage to healthy cells during radiation therapy. Importantly, the protective effects were confined to the injection sites, ensuring that tumors remained vulnerable to radiation. Future directions and implications. The implications of this research are vast. If successfully translated to human applications, this technique could greatly enhance the quality of life for cancer patients by mitigating the side effects of radiation therapy. Furthermore, researchers anticipate that the Dsup protein could also protect astronauts from cosmic radiation during prolonged space missions, underscoring the versatility of this approach. Next steps include refining the Dsup protein to reduce any potential immune response in humans and optimizing the delivery system for better efficacy. The researchers are also exploring the potential of Dsup to protect against DNA damage caused by chemotherapy, broadening its therapeutic applications. Differing perspectives from media sources. Coverage of the study varied slightly among different outlets. MIT News emphasized the broader implications for cancer therapy and astronaut safety, while Genetic Engineering and Biotechnology News highlighted the relative safety of mRNA delivery compared to DNA. The University of Iowa's reports focused on the unique approach to protecting healthy tissues and the potential for optimizing radiation therapy. Conclusion: a new era in cancer treatment. The integration of tardigrade-derived Dsup protein into therapeutic strategies represents a significant breakthrough in cancer treatment. This innovative research not only offers hope for improved patient outcomes in oncology but also opens doors to new avenues in space exploration and longevity treatments. As the research progresses, The potential to harness nature's protective mechanisms promises to transform the landscape of medical treatments for a range of challenging conditions. References: 1. MIT News, Tiny Tardigrades Protein May Help Cancer Patients Tolerate Radiation Therapies, /news.mit.edu/2025 Tiny Tardigrades Protein May Help Cancer Patients Tolerate Radiation Therapy 0226. 2. Genetic Engineering and Biotechnology News, Tardigrade Protein Protects Healthy Cells from Radiation Damage, www.geningnews.com/topics/cancer/Tardigrade Protein Protects Healthy Cells from Radiation Damage/. 3. University of Iowa News, Tardigrade Protein May Help Protect Cancer Patients from Radiation Side Effects, /radiationoncology.medicine.uowa.edu/news/2025/02 Tardigrade Protein May Help Protect Cancer Patients Radiation Side Effects. The recent research on the tardigrade-derived protein Dsup presents several counterintuitive insights that challenge conventional perceptions about radiation therapy and cellular protection. Here are some of the key counterintuitive points identified in the content. 1. Harnessing a protein from an extremophile, tardigrades. Known for their ability to survive extreme conditions, are often viewed as curiosities of nature. The breakthrough lies in utilizing a protein from these organisms to mitigate the damaging effects of radiation on human cells. This raises questions about the potential of extreme survival traits in other organisms being applicable to human medicine, suggesting that nature's adaptations can provide solutions to human health challenges. 2. Temporary expression over permanent modification. The research highlights the use of messenger RNA mRNA to temporarily produce the Dsup protein in human cells rather than directly modifying the cells with a foreign protein. This approach is counterintuitive because traditionally many therapies focus on long-term modifications such as gene editing. The temporary nature of this expression aligns with safety concerns, as it minimizes the risk of long-term alterations to a patient's genome. 3. Localized protection versus systemic effects. A significant aspect of this research is that the protective effects of the Dsup protein are localized to the injection site and do not extend to tumor cells. This challenges the assumption that protective mechanisms would also shield cancer cells from treatment. It suggests a sophisticated level of control that allows for enhanced cancer treatment effectiveness while protecting healthy tissues, which is a nuanced approach not commonly seen in traditional therapies. 4. Potential for broader applications beyond cancer. While the primary focus is on improving cancer therapies, the implications of using the Dsup protein extend to protecting astronauts from cosmic radiation and possibly reducing DNA damage from chemotherapy. This versatility is counterintuitive because it suggests that a single protein could play multiple roles in very different contexts, cancer therapy and space exploration, where the conditions and challenges faced are drastically different. 5. Innovative delivery systems. The use of polymer-lipid nanoparticles for mRNA delivery is a novel strategy that might not be immediately intuitive. The combination of polymers and lipids to enhance delivery efficacy challenges the traditional views on drug delivery systems, which often rely on more established methods. This innovative approach suggests that combining different materials can yield better outcomes than using them separately, highlighting a need for continuous re-evaluation of delivery mechanisms in therapeutics. 6. Focus on healthy tissue protection. The study emphasizes protecting healthy tissues during radiation therapy, which is often overlooked in cancer treatment discussions. The prevailing narrative in oncology tends to focus on maximizing tumor kill rates, sometimes at the expense of patient quality of life. This research shifts the focus towards improving patient experiences and outcomes by minimizing adverse effects, a perspective that may not align with conventional aggressive treatment paradigms. 7. Potential immunogenicity concerns. While utilizing the Dsup protein, researchers are aware of potential immune responses that could arise from introducing a foreign protein. The plan to develop a human-compatible version of Dsup to avoid immune reactions is a counterintuitive step, as many therapies prioritize immediate effectiveness over long-term compatibility and safety. This foresight could redefine how biologic treatments are developed, prioritizing patient tolerance alongside efficacy. 8. Implications for future research directions. The findings pave the way for further exploration into other proteins with similar protective functions. This is counterintuitive in the sense that it suggests that even more complex biological systems like those found in tardigrades may hold keys to solving human health issues, encouraging researchers to look beyond established models and consider biological innovations that are currently underexplored. In summary, the research on the Dsup protein from tardigrades not only highlights a remarkable scientific advancement but also invites a re-evaluation of established norms in cancer treatment and cellular protection strategies. The counterintuitive elements presented in this study reflect an innovative approach to leveraging natural mechanisms for human health benefits, potentially transforming the landscape of therapies in oncology and beyond. The groundbreaking research on the Dsup protein from tardigrades presents several significant insights that are likely to be overlooked by many. While the high-level implications of improved cancer therapies and astronaut safety are often highlighted, there are nuanced details within the study that underscore its transformative potential. 1. Mechanism of action and specificity. One of the most crucial aspects of the Dsup protein is its mechanism of action in reducing DNA strand breaks. The study highlights how Dsup binds directly to DNA in action that is fundamental to its protective capabilities. This specificity allows for targeted protection of healthy tissues while leaving tumor cells vulnerable to radiation therapy. This dual action is essential, as many existing treatments either fail to protect healthy tissues or inadvertently shield tumors. Diminishing treatment efficacy. Understanding this mechanism not only emphasizes the innovative nature of the approach but also lays the groundwork for future therapeutic strategies that could harness similar protective mechanisms. 2. mRNA delivery system. The research team developed a sophisticated polymer-lipid nanoparticle system for the delivery of mRNA encoding the Dsup protein. This method is significant for several reasons. Safety profile. The use of mRNA instead of DNA is a critical detail. mRNA transfections are generally transient and do not integrate into the host genome, reducing the risk of long-term genetic alterations and immune responses associated with DNA-based therapies. This characteristic is often underestimated in discussions of gene therapy and cellular modifications. Localized protection. The nanoparticles were engineered to target specific tissues, oral and rectal, which is crucial for minimizing side effects. This localized delivery system ensures that only the tissues most affected by radiation therapy receive the protective protein, which is a significant advancement over systemic treatments that might affect the entire body. 3. Clinical implications beyond cancer therapy. While much of the conversation around Dsup focuses on its application in cancer treatment, the broader implications for longevity and space exploration are noteworthy. The potential for Dsup to be adapted for protecting astronauts from cosmic radiation could revolutionize long-term human space travel. This aspect is often overshadowed by the immediate medical applications but represents a critical intersection between biological research and the future of human exploration beyond Earth. 4. Potential for enhancing current therapies. The researchers suggest that the Dsup protein's protective qualities could extend beyond radiation therapy to other DNA-damaging treatments, such as chemotherapy. This potential cross-application is significant as it could lead to the development of a new class of adjunct therapies designed to enhance the efficacy of existing treatments. The ability to improve patient tolerance to chemotherapy could lead to better adherence to treatment regimens and ultimately improved survival rates. 5. Future research directions. The study not only presents current findings but also outlines future directions for research, such as developing a human-compatible version of the Dsup protein to minimize immune responses. This forward-thinking approach highlights the researchers' recognition of the complexities involved in translating laboratory results to clinical applications. The emphasis on scalability and manufacturing processes is also crucial, as these factors often determine the feasibility of new treatments reaching the market. 6. Broader applications and environmental considerations. The implications of utilizing a naturally occurring protein for medical purposes raise discussions about bioinspiration in therapeutic development. The research encourages a paradigm shift towards utilizing nature's mechanisms to solve human health problems. This perspective can spur further research into other extremophiles and their potential contributions to medicine, pushing the boundaries of what is possible in therapeutic interventions. Conclusion. The Dsup protein's ability to protect human cells from radiation-induced DNA damage is a remarkable scientific achievement with far-reaching implications. The study encompasses not only the immediate benefits for cancer patients but also opens avenues for advancements in space exploration and the development of new therapeutic strategies. A deeper understanding of the protein's mechanisms, delivery methods, and potential applications could pave the way for transformative changes in how we approach cancer treatment and beyond. The significance of this research lies not just in its immediate findings but in the broader conversation it initiates about harnessing nature's innovations for human benefit. 1. Mechanism and efficacy of Dsup protein in radiation protection. The Dsup damage suppressor protein derived from tardigrades has demonstrated a remarkable ability to bind to DNA and mitigate radiation-induced damage. Research findings reveal that this protein can reduce DNA strand breaks by 50% in tissues exposed to radiation, particularly in areas vulnerable during radiation therapy such as the mouth and rectum. This localized protection enables effective tumor treatment while safeguarding healthy cells, thus addressing a significant limitation in radiation oncology. The studies focus on utilizing messenger RNA mRNA to induce temporary expression of the Dsup protein rather than direct protein administration helps avoid potential immune reactions, making it a safer and more effective strategy for protecting normal tissues during radiation therapy. 2. Innovative delivery system for mRNA. The research team developed a sophisticated delivery method using polymer-lipid nanoparticles to transport mRNA encoding the Dsup protein to specific tissues. This dual-component system maximizes transfection efficiency and allows for targeted delivery, ensuring that the protective effects are localized and do not compromise the effectiveness of the radiation treatment on tumors. This innovation represents a significant advancement in the field of nanomedicine and underscores the importance of optimizing delivery mechanisms for therapeutic applications. The successful implementation of this technology could set a precedent for future mRNA-based therapies in various medical fields. 3. Broader implications for cancer treatment and longevity. The application of Dsup protein technology extends beyond cancer therapy, with approximately 60% of cancer patients undergoing radiation therapy experiencing debilitating side effects. The potential to improve patient quality of life is substantial. Furthermore, the research opens avenues for developing real-world longevity treatments and protective measures for astronauts against cosmic radiation during long space missions. The versatility of the Dsup protein and its delivery system could lead to more effective therapeutic strategies not only in oncology but also in aerospace medicine and possibly in reducing DNA damage from other treatments such as chemotherapy. 4. Future research directions and clinical applications. The study's authors emphasized the need for further research to create a human-compatible version of the Dsup protein that minimizes immune responses and to enhance the duration and effectiveness of protein expression following mRNA delivery. Future studies will focus on scaling up the manufacturing process and addressing the immunogenicity of the Dsup protein to ensure its safe application in humans. The potential for this technology to transform radiation therapy, allowing for higher doses with reduced side effects, could significantly improve treatment outcomes for cancer patients. Additionally, the exploration of Dsup in protecting other normal tissues from DNA-damaging therapies highlights its promise in broadening the scope of its clinical applicability. 1. The discovery of penicillin. 1928. The discovery of penicillin by Alexander Fleming marked a monumental breakthrough in medical science. Fleming noticed that the mold Penicillium notatum produced a substance that killed bacteria. This serendipitous finding led to the development of antibiotics, drastically changing the treatment landscape for bacterial infections and saving countless lives. Penicillin's introduction in the early 1940s revolutionized surgery, childbirth, and the treatment of infectious diseases, highlighting the profound impact of biological discoveries on human health. 2. The development of insulin therapy. 1921. The discovery of insulin by Frederick Banting and Charles Best transformed the treatment of diabetes. Before insulin, diabetes was often a fatal disease. The ability to extract insulin from the pancreas of animals and administer it to patients allowed those with diabetes to manage their condition effectively. This breakthrough not only improved the quality of life for millions of diabetics but also paved the way for ongoing research into diabetes management, including advancements in insulin delivery systems and the development of synthetic insulin. 3. The introduction of mRNA vaccines. 2020. The rapid development of mRNA vaccines against COVID-19 by companies like Pfizer-BioNTech and Moderna represents a significant advancement in vaccine technology. This approach uses messenger RNA to instruct cells to produce a harmless piece of the virus, prompting an immune response without using live virus. The success of mRNA vaccines in combating the pandemic has opened new avenues for vaccine development against other diseases, including cancer and other infectious diseases, showcasing the potential of mRNA technology in modern medicine. The recent breakthrough involving the Dsup damage suppressor protein derived from tardigrades heralds a transformative era in both cancer therapy and space exploration. As research progresses, the implications of this discovery are vast, promising healthier cancer treatments, extended space missions, and advancements in longevity therapies. Below are several key predictions for the future stemming from this exciting development. 1. Enhanced cancer treatments. The ability to use the Dsup protein to protect healthy cells during radiation therapy represents a major leap in oncology. By minimizing side effects, patients will likely experience improved quality of life during treatment. This could lead to increased treatment compliance. Patients may be more likely to complete their radiation therapy when side effects are mitigated, potentially improving overall treatment outcomes and survival rates. Higher radiation doses, with the protection of healthy tissues. Oncologists may confidently administer higher doses to tumors, enhancing the efficacy of radiation therapy while reducing collateral damage to surrounding healthy cells. Broader application in chemotherapy. As research continues, the Dsup protein could also provide protective benefits against DNA damage caused by chemotherapy drugs, expanding its utility in cancer care. 2. Space exploration applications. The application of Dsup in protecting astronauts from cosmic radiation is another exciting prospect. As space missions extend, such as those planned for Mars, the risks of radiation exposure become increasingly critical. The following advancements are anticipated. Safe long-duration missions. Utilizing Dsup to shield astronauts can make long-term space travel safer, allowing for extended missions without the debilitating effects of radiation exposure. Development of radioprotective protocols. NASA and other space agencies may implement protocols based on Dsup to prepare astronauts for prolonged exposure to the harsh conditions of space. Fostering a new era of interplanetary exploration. 3. Advancements in longevity research. The Dsup protein's ability to protect DNA from damage could also lead to breakthroughs in longevity research. As the scientific community explores ways to combat aging at a cellular level, the following trends may emerge. Innovative anti-aging therapies. The principles derived from Dsup could inspire new therapies aimed at reducing cellular damage associated with aging, potentially leading to increased lifespan and improved health span. Personalized medicine. as researchers continue to refine the use of Dsup. Personalized therapies tailored to an individual's genetic makeup and health profile could become commonplace, further enhancing longevity treatments. 4. Improved mRNA therapeutics. The innovative use of mRNA technology to deliver the Dsup protein opens the door for a broader application of mRNA in medicine. Future developments may include targeted mRNA therapies. The success of Dsup delivery may pave the way for targeted mRNA therapies for various diseases, including genetic disorders and other forms of cancer, solidifying mRNA's role as a cornerstone of modern therapeutics. Regulatory frameworks for mRNA treatments. As mRNA technology becomes more prevalent, regulatory bodies will need to develop comprehensive frameworks to ensure safety and efficacy, potentially accelerating the approval of new treatments. 5. A broader impacts on biotechnology. The integration of natural proteins like Dsup into therapeutic strategies highlights the potential of bioinspiration in biotechnology. This could lead to novel therapeutics derived from nature. Researchers may increasingly turn to nature for inspiration, discovering new proteins and mechanisms that could be harnessed for medical applications, thereby expanding the arsenal of available treatments. Collaborative research ventures. The interdisciplinary nature of this research may foster collaborations between biologists, engineers, and medical professionals, leading to innovative solutions to complex health challenges. Conclusion. The integration of the Dsup protein into therapeutic strategies represents a significant breakthrough with the potential to reshape cancer treatment, enhance the safety of space missions, and pave the way for longevity therapies. As research progresses, the implications of this work will likely extend far beyond oncology, influencing a wide range of medical fields and fundamentally changing our approach to health and disease. The future appears promising, with the potential to unlock new frontiers in both human health and exploration beyond our planet.